Hello everyone. Today's topic is transpiration. Transpiration is a process in which plants lose water to their environment through their leaves in the form of water vapor. Procedure presented in this video will measure the rate of transpiration under given conditions. And later applications may involve measuring the rate of transpiration under the influence of different variables such as light intensity, humidity, temperature, number of stomata, and etc. Transpiration is a pretty important concept in plant physiology because it is one of the three forces that are hypothesized to explain water transport by plants to the parts such as leaves that may be located 100 meters above ground. Now we're going to go ahead and start the experiment. We will need a plant cutting from a fresh plant, a razor blade for cutting plants, a plastic tubing in order to connect plant cutting to gas pressure sensor, a gas pressure sensor in order to measure the pressure change in the plastic tubing, a Go-Link interface to connect gas pressure sensor into your computer, a ring stand and clamps to secure our setup, petroleum jelly to make a tight seal on plastic tubing, a bucket of water to make your cutting under the water, a syringe to fill plastic tubing with water, a pair of scissors to make cutting from plants. Cut off a piece of your plant using scissors. Place plant cutting and plastic tubing underwater. Inject some water using your syringe into the plastic tubing. We're doing this to make sure you have a continuous water column in the plastic tubing. Make another cut on your plant while it's underwater, about 1 inch from its end, and cut it with an angle. You can throw away the remaining piece. Place the plant cutting in the plastic tubing underwater. Make sure there is no air bubbles in the plastic tubing. If you think there are some bubbles, work it out until you see no air bubbles inside the plastic tubing. Push some water into the plastic tubing using syringe to make sure there is no air bubbles inside the plastic tubing. Using a clamp, secure the plant in the plastic tubing while it's underwater. Now I just put the clamp around the plastic tubing. I'll just push it on the ends to secure the plant. Install your clamps on the ring stand so you can set up your plant cutting and the sensor for your experiment. Connect your Go-Link interface into the USB port of your laptop. Connect your gas pressure sensor into the Go-Link interface that is already installed to your computer. Using petroleum jelly, seal the end of the plastic tubing where you have the plant, while the other part of the plastic tubing is still underwater. Closing the other end of the plastic tubing using your finger, remove your setup from water and set it on your clamp. Place your plastic tubing with the plant cutting on your setup. And the other end, and I'm going to connect the other end to the gas pressure sensor. And place gas pressure sensor on the clamp. This is how your setup should look like. After you're done, you use some paper towels, wipe off the excess water on the leaves, you start your Logger Pro software and go to File tab on your left hand side. Go down and see Open, click on it. You will see a lot of folders with a lot of presets. Now you go to the folder where it says Biology with Vernier, click on it, and you scroll down and select Preset for Transpiration and click Open. This is the preset for this lab, where you see the live measurement of pressure, which is the relative measure of transpiration, and on your left hand side you see the data table, on your right hand side you see a graph. When you click Collect, it will start data collection, which indicates the rate of transpiration by this plant. Data collection will last for 15 minutes and it will end automatically. Now our experiment is complete and data collection has stopped automatically, as you can see here. 15 minutes and data collection is complete. Now we're going to use this data to calculate the rate of transpiration. Data is plotted on this graph in the form of dots, but you see connected dots as a line graph. Now we're going to make a best fit line for those plotted data in order to calculate the slope of the line, which will give us the rate of transpiration. In order to do that, you go to the menu up here and click on this tab where you see this function with a best fit line and an R value. When you hover over it, it tells you that it's the linear fit. Click on it and it will automatically calculate some values and make a best fit line for your data collected. The calculations automatically made include the slope of the line and y-intercept along with correlation. Slope of the line gives you the rate of transpiration, and units for this is kilopascals per minute. You can convert this value into pascals per minute if you think this is a number that is too small to work with. Using this slope, using the rate that you calculated just here, you can make comparisons between the manipulations if you do so, such as changing the environmental conditions, including light intensity or humidity. Calculating rate will allow you to make comparisons. This is pretty much completing our experiment. Transpiration is a very important process, The environment around us is habitable because of the plant activity such as photosynthesis and transpiration. Water is lost to the atmosphere by transpiration at a way faster rate than it occurs through evaporation on the same area of water surface. That means most of the rain is brought to us by plant transpiration. Transpiration is also so important, especially if you are interested in growing plants. Well, everybody on earth actually is dependent on plants at least as source of food. And at least some people must know a lot about their physiology in order to supply humanity with enough food. It is also good to educate everybody about the basics of these processes to decrease their likelihood of making silly mistakes that can result in disturbing the environment too bad for our precious and delicious friends.